Welcome, everyone. And it's an honor to be here to talk about the center of our bioscience endeavor, that is the microbiome, and how we will continue to advance the information that has been gathered over time concerning the weeding, seeding, and feeding of your microbiome. I have no potential conflicts of interest relevant to this presentation, and the slides I'm presenting are my own, and those I've modified from what I view as some of the leading experts in this area, and I've referenced some of their presentations at the end of this presentation. I should emphasize that the content of this is designed for educational purposes and not intended to diagnose, treat, or prevent any specific infectious disease. And we always encourage you to work with your healthcare practitioner in making any changes in your medication, your nutritional plan, or overall health care plan. The outline of what I'm going to be presenting is on this slide, and this will basically kind of review some of the terminology, foundational principles that we reviewed in our last webinar. And then I'm going to kind of go through some of the aspects of weeding, seeding, and feeding the microbiome from the lens of a microbiologist and infectious disease specialist, getting into an area what I have referred to as a subspecialty now in infectious diseases called therapeutic microbiology. The follow will be Dr. Luby, who will present this topic from the lens of a functional medicine physician, since we are commonly in an era where we have a lot of dysfunction. How do we approach correcting this dysfunction from a functional medicine perspective, which he'll cover? We'll cover what is a healthy gut microbiome? What are the factors associated with an intestinal dysbiosis? What's the significance of gut dysbiosis? And then finally, how do we manipulate the gut microbiome or biota to optimize health? We'll be focusing today on the human gut microbiome as we reviewed in the past. We have approximately 39 trillion bacteria in and on our body. 38 trillion of those are in the colon, and that's what we're going to be emphasizing today, that human gut microbiome primarily in the colon. And by a cell count, as you know, we're only about 43% human, and by the gene count, where there's 150 times more genes in the microbes than human, we're only about 1% human. Well, let's look at, again, recap some of the terminology. We'll be discussing microbiota. That's the updated term. We've gone away from microflora, which primarily in retrospect refer to the plants, but talking about bacteria, but this can also include viruses, fungi, and protista. These are the microorganisms that live in our ecosystem, and now today, specifically the gut, and primarily we're going to be talking about bacteria. The microbiome, the combined genetic material then of these organisms, and their genes, and dysbiosis, which will be the center of our discussion as far as the imbalance that can occur in the microbiota and how that affects our health. The metabolome then, finally the metabolites that these bacteria produce, genetic material as well as molecules that are being released. It's been estimated that anywhere from about 1/3 to 50% of the molecules circulating in our bloodstream as a human are derived from the bacteria in our gut. Well, this is a picture of the microbiome from the Sonnenberg lab in Stanford, kind of one of the prettiest pictures. And these little so-called the little jelly bean-like structures, and this is your microbiome. Again, in the colon, about 38 trillion of these circular blue things here are the colonocytes and nuclei of the colonocytes. And as you know, what divides us from the outside world and the microbiome to our inner self is this mucus layer. And as it's been said, fences make good neighbors. Well, the core functions of the gut microbiome: defense against pathobionts, so the pathogens such as, and starting with Helicobacter, the more pathogenic E. coli, Salmonella, Shigella, and so on. The gut microbiome helps maintain that gut barrier, the mucus layer. It helps digest so-called non-digestible nutrients. And as Alessio Fasano says, unlike Las Vegas, what happens in the gut doesn't stay in the gut. And indeed, the intestinal microbiota controls our entire body in ways, and it controls what our weight is. It controls if we have diabetes. It controls immune development. It has a significant impact on if we have autoimmune disease or not, or allergies like asthma. And it also has an effect on if we're depressed, anxious, or have other behavioral problems. Well, what is a healthy gut microbiome? Well, we're still on the hunt for a healthy microbiome, and the scientists are trying to figure it out. But this review by Renanella and colleagues gives us some hint of what a healthy gut microbiome may be. And it's centered around that there's no known universal normal microbiome. And we know we inherit this from our mother. In fact, in utero, it appears we are even starting to acquire bacteria, which hopefully are all beneficial. This is an extremely dynamic form of existence that dwells within us, and changes from individual to individual occur, and within the same individual over time as we age. Optimal healthy gut microbiota composition is different for each individual. We're all unique, but everything is connected, as we've implied, and everything matters, as we'll come back and discuss. Each individual is provided with a unique core gut microbiota profile that plays many specific functions, with the core gut microbiota functions being highly preserved between individuals. That means you and I have a different core microbiota potentially, but they're carrying out very similar functions that either maintain our health or make us sick. Richness and diversity of the gut microbiota is shaped early in life, and it's pretty well fixed by 3 years of life, as we now understand. And hopefully we gain a healthy gut microbiota composition. The richness referring to the abundance of a specific organism. Diversity can be divided into alpha diversity, which is the number of species which is in a given ecosystem, such as your gut, or beta diversity, the difference in species between different ecosystems, such as the mouth or the colon. A core native microbiota remains relatively stable in adulthood, and throughout life, the richer and more diverse the microbiota, it appears the better resistance and resilience we have to disease, including infectious disease such as SARS-CoV-2. We also now understand that there's keystone bacteria that play very important roles, and they don't necessarily need to be there in great abundance, but need to be there that kind of act as little bacterial engineers to keep us in balance. And then there's bacterial guilds, bacteria that form together. And there's been identified first, second, and third generation guilds. And when we're eating and feasting, they feast upon what we're eating. So you're feeding trillions every time you eat. When we go into a fasting state, the metabolites from that first generation are feeding the second generation. And the metabolites from the second generation during the fasting state are feeding the third generation. And then the metabolites from the third generation can feed back to that first generation to keep them going, to keep us healthy. There hopefully is an ongoing healthy host microorganism balance, the so-called superorganism that we now understand we live with. And this needs to be respected, as we'll be discussing, to optimize metabolic and immune functions and prevent disease development. Just briefly, again, through a microbiology lens, there's two major phyla, the Firmicutes, the Bacteroidetes, and these constitute probably more than 90% of the bacteria found in our intestinal tract. And this is a little bit noisy, but microbiologists love this type of slide. This is a taxonomic gut microbiota composition, the phyla, class, order, family, genus, species, and you can get down to even strains here that you can see in the middle here, Firmicutes, Bacteroidetes, again, greater than 90%. You'll recognize some of these. There's the Lactobacillus reuteri, that's a keystone organism, and recent studies have suggested that we have lost this species. Only about 1 in 20 Americans seem to have retained this species, and it plays a very important role in health. The other keystone here on this list is this fecal bacterium on the top there. In the Bacteroidetes, there's Bacteroides species, Prevotella, that play an important role in health. And then there's these other phyla as shown here, the actinobacteria, and you may recognize the bifidobacterium here at the top of the list, commonly found in probiotics. Now, this devious phyla here, the proteobacteria, may be healthy or maybe diseased, and that includes E. coli, Shigella, and so on, and Fusobacteria. And then the phyla here has another keystone organism called Akkermansia mucinophilia. And Akkermansia plays a very important role in maintaining that mucus barrier. And it's been shown that if you don't have that organism, you're not going to respond to immunotherapy for cancer. So it's important to bloom that out if you're getting immunotherapy. Well, what are the factors associated with intestinal dysbiosis? This is a slide that we presented the last time, and it just kind of goes through all the factors associated with loss of species, starting with what preceded us in terms of second or third generations, the antibiotics, the standard American diet, and all the so-called environmental pollutants are now what is a new area of study called the exposome, the environment, and how that impacts our disease, the diseases we acquire, or our health. So loss of species, dysbiosis of the gut microbiome, dysbiosis then, starting with the normal healthy intestine, we can lose beneficial bacteria, especially these keystone organisms. We can have overgrowth of potentially pathogenic bacteria. This includes in the small intestine, where we can have SIBO, or small intestinal bacterial overgrowth, or we can have SIFO, or small intestinal fungal overgrowth. And then finally, we can have loss of overall bacterial diversity. And we, over a lifetime, even starting in childhood sometimes with antibiotics or being born C-section or not getting breast milk, we can begin to lose the microbiome complexity and stability. Well, what's the significance of gut dysbiosis? It's summarized on this slide, that we now understand that a dysbiotic, imbalanced human microbiome is associated with a multitude of chronic diseases. In fact, maybe all. And this includes starting with the skin, where we have the so-called gut-skin axis, where there's bidirectional talking going on. Psoriasis is associated with this, acne, skin cancer, heart disease, atherosclerosis, heart failure. The gut-brain axis, it determines if we're depressed or anxious. Autism is linked with a dysbiotic microbiome. The gut-lung axis is important in determining, do we get pneumonia or not? Do we get asthma, chronic obstructive pulmonary disease? And it seems to be linked with lung cancer. And it affects our metabolism. It's linked with being overweight, type 2 diabetes, mitochondrial dysfunction leading to loss of energy, and then cancer. So we now understand why Hippocrates was correct. All disease begins in the gut. Well, how can we manipulate the gut microbiota? Can we repair our microbiota and improve health? Well, this article gives us a kind of a jump start in understanding where we're at with this kind of a revolution, a paradigm shift in understanding how we can turn all these good bacteria towards optimizing our health. And they outline in this review different strategies used to manipulate the gut microbiota and the prevention and treatment of disorders, and target microbiota dysbiosis, trying to move forward. Although with the complexity and the load of data, we know as we move forward, we've got a real challenge in front of us, as this cartoon implies. Well, this is a list then of the strategies we have for therapeutic microbiology: food, probiotics, prebiotics, symbiotics, or the combination of probiotics and prebiotics, realizing the probiotics need to live and they depend on prebiotics. There's postbiotics, and those are the metabolites produced by the bacteria. Sometimes we have to use selective antibiotics, like in the setting of SIBO, or small intestinal bacterial overgrowth. We have microbiota transplant, AKA, as it sounds, fecal microbiota transplant. And we've done over 100 of these at Methodist in the last few years, although we've had a pause because of COVID-19, specifically for CDI, or Clostridioides difficile infection. And then finally, we have new tools in our toolbox that are being explored: microbial engineering, microRNAs, hyaluronan, that substance in the extracellular matrix, and then some nanomedicine-based approaches, and then finally using CRISPR gene editing. Well, let's just take a brief look at probiotics. Michnikov popularized probiotics and first described this in the form of a fermented dairy yogurt. Since his introduction to this concept, there's been changes in how we define probiotics. In 2001, probiotics were defined as live microorganisms when administered in adequate amounts confer a health benefit on the host. And then this was complicated by a scientific committee in 2014 who concluded that evidence for a probiotic can be sufficient to support the concept of potentially beneficial live microbes for which some plausible benefits are reasonable to expect, including strains of microbes that have not been specifically tested for health benefits, but are members of a well-studied genera or species. In other words, bacteria that are friendly. They complicate this by some complicated definitions and criteria, which I'll let you go back and review. And basically it divided not probiotic, which many fermented foods are really just labeled as live or having active cultures without specific efficacy studies. But by and large, fermented foods, which we'll be discussing, have been labeled as probiotics, even though this committee decided they're not truly a probiotic. And then probiotics are divided based on the science supporting them, from a spectrum that there's no health claim to a specific health claim. And then there's at least one probiotic that is a so-called probiotic drug. Well, what do we know about the science supporting the use of probiotics? The Stanford team, Erica and Justin Sonnenberg, had developed a human microbiome science department, and they've been doing a number of studies. I'm just going to briefly go through some of those. One was looking at probiotics, addressing the question, can they improve the microbiome? Can they decrease inflammation? Can they reduce severity of the metabolic syndrome? Probiotics did not increase microbiome diversity. They didn't affect the inflammatory cytokine levels, as it turns out, when they randomized approximately 36 patients. They didn't improve the parameters of metabolic syndrome. However, when they put this information all in a computer and analyzed the food, the nutritional plans these patients were taking in, with the placebo group up here, each row serving as an individual, and there were two distinct groups in the probiotic, with the row again, each individual participant, and then the columns representing the various metabolic parameters, with red being actually decreasing benefit and the brighter and darker blue purple representing benefit. And as you can see, there was this subgroup that had decreased triglycerides, decreased liver enzymes, or decreased fatty liver, and decreased insulin resistance. And the question was, well, why is that? Well, they went back and analyzed the computer data, and it turned out responders to probiotics were eating more plants, vegetables, fruits, grains, legumes. Well, this slide just kind of briefly outlines guidelines for selecting probiotics. I'll let you go back and review that. I often, when patients ask me, well, what probiotics should I be taking? We sit down and look at the consumerlab.com website. This is a science-based website, and they go through the various uses of probiotics and the science behind it. So we'll look at, maybe I have a patient that's kind of anxious and depressed. Well, I go down and we look at, there's some evidence now supporting two bacteria, Bifidobacterium longum and Lactobacillus helveticus, help reverse anxiety and depression for certain patients. And so I send them down the street a couple blocks away, and they get a mood probiotic. And again, with some science supporting that. Well, how about prebiotics? The Consumer Lab report also has some review of prebiotics. The scientific committee that we already referred to defines a prebiotic as a substrate that is selectively utilized by the host microorganisms, conferring a health benefit. They go through this algorithm, as you can see in the center here. Dietary fiber is the key prebiotic. And as time allows and as interest leads us to discussing there's soluble fiber, there's insoluble fiber, and there's a resistant starch that all makes up the dietary fiber, with the less fermentable, which by their definition is not truly a prebiotic, is the non-soluble fiber. Well, how about food? And this will lead us into Dr. Luby's presentation, that we now understand food may be the main way, the central way of manipulating our microbiome as a therapeutic microbiology intervention. And if we glance at the prebiotics that are found in foods, you'll see a number of foods, vegetables, especially some of the grains here have prebiotic, inulin, oligofructose. We also know diets rapidly and reproducibly alter the human gut microbiome. And in this final study out of Stanford that looked at the gut microbiota targeted diets modulating human immune status, or the so-called Phi-Pho study, where they randomized individuals into a high fiber diet or a high fermented diet. The high fiber diet, their participants started out with less than 20 grams of fiber, and they had a ramp up phase over 4 weeks, and then 6 weeks of eating increased fiber of over 40 grams, vegetables, fruits, grains, legumes, which they regarded as prebiotics. And then there was a group they randomized to fermented foods, which I'm sure everybody munches on every day. The original GT, kombucha there, yogurt, kefir, kimchi, sauerkraut. And they were able to ramp up from a baseline of only a half a cup of fermented foods up to 7 fermented helpings every day in this group they studied. And they loosely referred to this as probiotics. But what were the results? Fiber, microbiota diversity, no change. Inflammatory markers, no change. Fermented foods, a significant increase in microbiota diversity. And inflammatory markers decreased by 20%. Now, to be fair, those that were getting the fiber, there were some other parameters they looked at. Indeed, there was a blooming out in abundance of the microbes that feasted on the fiber. That was a benefit. And then there were certain metabolic benefits. There were decreased substances or molecules that potentially are deleterious to our health. So there was some benefit in the background, even in the whole group when they looked at it. Now, it was interesting when they looked at the fermented foods and the new bacteria, they went out and purchased all these fermented foods and looked at and logged in all the bacteria. And indeed, when they said live and active cultures, they meant it, because they were able to culture out all these different bacteria. Now, if you look off to the right here, this circle, this little sliver, that was the 5% of the new microorganisms they encountered in their participants. They went from a baseline of about 100 to acquiring over 25 new organisms. But however, it turned out only 5% of those new organisms came from the fermented foods, and 95% of the new species of bacteria were not found in the food. And they were really left pondering that. So Phi-Pho results, fermented foods, consistent benefit across all participants, although a somewhat limited pilot study, but still very significant. The New York Times took this up actually in 2 separate articles. Fiber, individual differences they found, and when they broke down what these individual differences were, they found that individuals that were in the high fiber group, if they came into the study with a pre-existent baseline increased alpha diversity, in other words, the increased number of species, they did get a benefit, decreased inflammation, versus those that came in with a low diversity, they actually had increased inflammation. And the two together canceled out one another for the group effect. So fermented and fiber foods do different things. We know a food such as broccoli can't be digested by our enzymes, but can go to the colon, and bacterial enzymes can degrade the soluble, insoluble fiber or resistant starch that passes down. And there's more microbes that feed on those specific fermented foods or fiber foods. I should say better function and lower inflammation if your diversity is high, versus the fermented foods, overall higher diversity, lower inflammation, and probiotics work best in the context of a better diet. So final take-home messages, not all microbes are bad. In fact, most are good. Indeed, a paradigm shift in microbiology and infectious diseases. Fermented foods appear to compensate for our sanitized environment. Fermented foods improve the microbiome and immune system. Probiotics may be beneficial in the context of a healthy diet, and blending high fiber and fermented foods may provide the greatest benefit. So Erica Sonnenberg provides you these recipes for blending fiber and fermented foods: yogurt with nuts and berries, vegetable miso soup, hummus, lentil, and sauerkraut salad, kimchi fried rice, kale and kefir smoothie. Yummy. And now we know our food should be our medicine, and our medicine should be our food. Thank you. And there's references that follow. And then again, I provided some links to the various reference presentations from various experts. Thank you.
Thank you, Rachel. And Dr. Penn, what a powerhouse presentation that was. So thanks for that. Welcome, everybody. For those of you who got to know me in the earlier presentation, welcome back. For those of you who are new this time, good to meet you virtually for the first time. And I'd like to emphasize how Dr. Penn ended here. You know, if you think 200 years ago, or whenever we first discovered bacteria, over time, as we came to understand what they do, they were considered probably the greatest threat to human health, and maybe even as recently as 20 years ago, the greatest threat to human health. And now we understand that bacteria are the greatest benefit to human health if we take care of them well, which is why our mantra at IFM is weed, seed, and feed. And what I'm going to do here is take you away from the science a little bit and really focus on your role in the healthcare team. Many of you do not actually initiate the therapies. You don't initiate the probiotics, the prebiotics, et cetera, but you will be communicating with patients, interacting with patients who are attempting to do that, whose primary clinicians have actually put them on some kind of a regimen with probiotics or prebiotics or a dietary plan that incorporates all this. So we're going to talk here a lot about some practical aspects of communicating with patients around this. On that note, weeding, seeding, and feeding. What does that sound like? If you did not know this was a healthcare lecture, you might think this was a master gardener's course, right? And that's a good leverage point for patients. If they don't understand, it's hard for patients to get their mind around this fact that, oh my gosh, we have all these bacteria in us. I say, yes, we're not really a single human being. We're actually an ecosystem. We're a lot like a garden. Those are the analogies I use. I think those really resonate with patients because so many of your patients will like to garden. So many of your patients will have an understanding of an ecosystem. Another communication technique I find effective is to ask your patient, where's your favorite place to go in nature? And most likely, they're not going to say a soybean field in Wisconsin or a cornfield in Nebraska. They might say the Lorenzen Gardens in Omaha, but why do they say that? And the place they choose is going to be a place of high diversity, most likely. High species diversity, both with flora and fauna. And then you can tell them that's what you want your gut bacteria to look like. You want a lot of diversity there. And you want to take care of that like you'd take care of your favorite place in nature that you go to, or like your vegetable garden or your flower garden in your yard. That's how you want to care for your own microbiome. And then women always resonate with the idea, if they've been pregnant before at least, of you're not eating for yourself, you're eating for two. Well, as Dr. Penn said, you're not eating for yourself, you're eating for billions or trillions. And that is so true with the microbiome. So let's weed, seed, and feed as we talk about it in the functional medicine training programs, with a special emphasis on your role in the healthcare system and how you can communicate with patients. This is the anchor slide that you'll keep seeing over and over. And let's focus first on weeding. Weeding implies that we need to get rid of something in the gut or get rid of something in the patient's behaviors, perhaps. And one thing is certain foods. Dr. Penn mentioned certain foods that are very good for the microbiome. There are some foods that can be adverse or can have adverse effects on the microbiome. This is why an elimination diet can be so important. Some foods cause sensitivities in certain patients, which are immune mediated, not necessarily allergies, but sensitivities which can cause inflammatory symptoms. And then there's other foods which are simply straight out inflammatory that can hinder the growth and the diversity of the microbiome in your gut. It's important for you to understand how many different kinds of contributors there are to derangement in the microbiome, or that word dysbiosis. So this is something you can convey to your patient. It's not only nutrition, it's lifestyle factors, it's stress levels, it's other conditions that they may have, it's other medications they may take, it's toxin exposure. So personal care products are a very important factor there. So important for you, again, to convey the wide spectrum of influences in a person's life that can affect their microbiome, and not just to focus on food or supplements. It's important also to understand, again, thinking back to when we thought bacteria were the greatest threat to our health, that what we're talking about with a dysbiosis condition, it's not a true infection. It's, as Dr. Penn's slide showed, a derangement or an imbalance. And with that in mind, you can reassure your patients, you don't have to jump to pharmaceutical antibiotics. What you want to do first is the dietary changes and the pre and probiotics, and sometimes postbiotics or symbiotics. If you have a condition of dysbiosis that is not going to improve with that initial therapy, then we consider antimicrobial botanical agents and antimicrobial medications, pharmaceuticals, in other words. The next few slides are kind of busy, but I'm going to show for you clinicians just exactly what we recommend in what situations therapeutically and according to what lab test results you might have. There are stool lab tests that can give you a fair idea of the diversity of the bacterial species and what they're producing in terms of metabolites. So if we're talking about bacteria, and in the first column there, if your stool test shows elevated pathogenic bacteria or potentially pathogenic bacteria, prebiotics and probiotics are almost always a good intervention. Dietary changes, of course, dietary changes are going to be across the board here, so you just want to keep that in mind. What else to consider? If it's a true infection as opposed to a dysbiosis, you go with antimicrobial therapy. And also if the signs and symptoms would indicate that, if it really looks, fever, severe diarrhea or something, then it's something beyond just a run-of-the-mill dysbiosis. If the organism that's detected is a yeast or a candida, then it's very important in the dietary part of your plan to have a low-carb diet and a low-sugar diet. Here's where you can really help your patients. A lot of patients don't understand what a carbohydrate is. What we're really speaking about here is refined carbohydrates from grain sources or just pure sugar and or sugar substitutes. So what you wanted to do is you actually want to have a diet that's rich in complex carbohydrates coming from plants, especially plants with fiber, as Dr. Penn mentioned. But you really want to cut out the low carb, the high refined carb foods. And that includes many baked goods, anything that uses flour, sugar, butter, for example, our favorite combination when we bake. If some of you remember when I was speaking about in the last webinar, how do you know a plant food has a lot of fiber in it? Well, it's the parts of the plant that are above the ground. Remember, they have to resist gravity or attract insects or gather sunlight through photosynthesis. So it's the stems, the leaves, and the flowers of a plant. High carbohydrate parts of a plant that have a lot of starch or a higher glycemic load that might need to be reduced in a anti-yeast diet would be the flesh of fruits. That's rapid energy for the seeds. And then the flesh of roots, tubers, potatoes, all of the parts of the plant that grow underground can be high in starch and more readily available sugars that could be used by yeast. So those are some things that might need to be avoided if a person has yeast overgrowth. Probiotics are very helpful, especially one called Saccharomyces boulardii. And then fairly often you'll need to use antifungal agents. There's botanicals and there are pharmaceuticals. When you have to take them by mouth, a pharmaceutical prescription is guided by the severity of the condition and usually reserved for the severe cases or the recalcitrant cases. Parasites, if those are found on a stool test, those often do need to be treated right away with botanicals or pharmaceuticals. Again, depends on the clinical context. So that's weeding. How about seeding? This refers to giving the gut probiotics, giving the gut organisms, the microorganisms, and sometimes it's even been found now, if they're not live, they can still be beneficial. What we teach our clinicians is use probiotics in food sources first. This reemphasizes what Dr. Penn said about fermented foods. So here's a place you can really help your patients. If they can't make their own fermented foods, I like to make kimchi. I've always got a crock of kimchi going in my kitchen, but there's many products in the store, and I'll show you a slide, and we've got a toolkit item that you can show patients. There's many items in a store that they can incorporate into their diet, and you can mix these in with other foods if they don't like the taste particularly. You saw Dr. Penn's kimchi fried rice can be a great way to go. Put some maybe sesame oil on it, a little soy sauce or something. It can be a delicious meal. Supplements, I won't cover that here in depth. Dr. Penn really covered that. The duration, often 6 to 12 weeks is a rough guide, but there is no absolute duration that you give these. It depends on the patient's condition and their response. In some cases, probiotics can be continued indefinitely, and certainly fermented foods can be eaten on a daily basis. Here's a list of certain probiotic-rich foods, the ones on the left being the most well-known, the most available, the ones on the right either being less available or having some issues, like kombucha starts with a lot of sugar when you're making that. The beers, they have their own issue with if it's an alcoholic beer, but there's some carbohydrate there as well. And then sourdough, that does have some refined carbohydrate, but it does contain some, it's more beneficial if you have to have a bread product than a non-sourdough bread, for example. The toolkit item that you have access to is this one, the probiotic and prebiotic foods. IFM has developed this and many other dietary and other kinds of toolkit items like this. So this is one you have access to, I believe, in your portal or in your materials here. You can use this with patients. It can be a shopping list for them. Really good for them just to take this to the store. Okay, so much for seeding. I think the most important thing is feeding. And now if we're thinking about your body being a garden, this is similar to fertilizer in the soil or putting compost, mixing compost in with your soil. That's what feeding does. And this microbiome in your gut does need to be fed. So as Dr. Penn said, what you are eating is the primary thing. You're eating not just for yourself, you're eating for trillions. So you do want to eat the foods that they like to consume, but you can also supplement that with certain prebiotic supplements. So Dr. Penn went over the definition of a prebiotic, but it's a substance that's used by these bacteria to benefit them and for them to provide us with even more beneficial metabolites in return. It's a great bartering system, so to speak. Some of those metabolites that they produce are called short-chain fatty acids. And even though it has the word fatty in it, fatty acids, it's a very beneficial group of metabolites. You can see on the right here, the most important ones are butyric, acetic, and propionic. Butyric acid, or butyrate, has probably had the most research and is most widely recognized as the short-chain fatty acid that confers the most health benefits. So if you get lab tests that measure your butyric acid or butyrate, you want to see high levels of that. So back to our therapeutics. Prebiotics can be incorporated into dietary protocol unless contraindicated. There's only a few patients who might react poorly to prebiotic foods that contain high amounts of fiber. The small intestinal bacterial overgrowth might be one condition where a diet that is very high in fiber might be harmful at first. Wait until you fix that small intestinal bacterial overgrowth condition first. Prebiotics, we almost always recommend them to be accompanied with probiotics or fermented foods. Again, 6 to 12 weeks is a common duration, but these can be given indefinitely. And again, every individual should be eating prebiotic foods every day, those plant-based foods that Dr. Penn talked about. The benefits of butyrate are many. This gets back to Dr. Penn's concept of what happens in the gut does not stay in the gut. The butyrate certainly helps the colon, and it's responsible for the repair and regeneration and maintenance there, but it reduces inflammation and oxidative stress, and that has effects throughout the body. And those initials down there, BDNF, GDNF, NGF, those are involved with neurological function anywhere in the body. Proteins and enzymes activation, that's all throughout the body. So these live microorganisms in our gut really do have systemic effects. It is not Las Vegas, fortunately. What happens in the gut does not stay in the gut. You can help almost any health condition of your patients by helping them improve the health of their microbiome. Specifically, prebiotic supplements and prebiotic foods can have these specific effects. I'll let you use this with your patients as a reference slide. And I just want to close out by reinforcing the idea that our body, our human body, is a beautiful thing. It's like a garden. And the more diversity, the better. Just as we love to see the more different kinds of flowers, the better. The more different vegetables growing in our garden, the better. And it's like our favorite place in nature. We need to be wildlife managers because our favorite places in nature usually have a wide diversity of plants and a wide diversity of animal life. And I'd like to tip my hat to Julie Segre of the NIH, who said, we need to be microbial wildlife managers of our own bodies. With that, we'll turn it to question and answer session. So I turn it back to you, Rachel.
Wonderful. Thank you so much. Again, wonderful presentation, a lot to think about as well. So we are going to be as interactive as we can with our virtual audience here. So I'm going to share my screen and we are going to get started with a Kahoot game, which many of you have probably played in the past. So, all right, can you see my screen, Dr. Penn? I can see you on there. You want to give me a thumbs up if you can see it? Perfect. All right, so pull out any device that has Wi-Fi. It could be your smartphone, tablet, whatever connects to Wi-Fi is all you need to play along. I'm going to give you this Kahoot game number in just 1 minute, but after you go to kahoot.it, so KAHOOT.IT, you're going to be prompted to enter a game pin number. And I will do that in just a moment. It's loading, so just a second. All right, so your game pin number is 4966965. Again, that is 4966965. And I'm going to minimize that for just a moment. I want to share with you. When you are there on that Kahoot, you can enter whatever name you want. It can be a nickname, it can be your favorite place to travel. It doesn't matter because we're not playing for any awesome prizes here. We're just playing for fun. Well, I think maybe we'll kick it off with that question about, are there certain illnesses, diseases, things like that, that people do have to be careful of? If you guys, Dr. Luby or Dr. Penn, want to jump on that one, I can start with that.
Well, the specific question was about thyroid conditions, and there's something to be said about that. There are some foods which are known as goitrogens, or foods that may have an adverse impact on thyroid function. And if you look into the medical literature and the research on this, it's not real robust, I would say. So I think it's very individual. Now, the other side of that is nutrient deficiencies also adversely affect thyroid function. So if you're not eating a good, diverse plant-based diet, you're unlikely to, or you're actually very likely to be harming your thyroid function by virtue of having nutrient deficiencies. If you're eating a highly processed diet, you're likely to have thyroid problems. And thyroid hormone production and conversion from the inactive to the active form both depend on an absence of dysfunctional inflammation. So if a patient of yours, or if you are eating a pro-inflammatory diet, again, a lot of processed foods, a lot of sugar, refined carbohydrates, you're going to be harming your thyroid function. So yes, there may be something to some goitrogenic foods. I guess especially some of them, if they're undercooked or uncooked, can be a bigger factor. But I think that's very individual. The larger factor, and by far the more common problem, is the pro-inflammatory diet that impairs thyroid function. And that's where you can make huge gains with patients who have thyroid disease. You can improve their production of thyroid hormone, their conversion of thyroid hormone, and even their cellular sensitivity to thyroid hormone by improving the diversity and the really phytonutrient content of their diet.
I could add to what Dr. Luby has stated. I should reflect, he's the expert in this area. I've heard him give expert lectures on this very subject. So he knows what he's talking about. I could just add, the patients I see, we commonly will screen when they come in with medically unexplained symptoms. We'll screen for the most common autoimmune condition out there, and that's Hashimoto's thyroiditis. It's not well known, but I've heard up to 95% of hypothyroidism is Hashimoto's. And then any autoimmune disease is kind of that three-legged stool: genetically predisposition, gut dysbiosis is commonly the environmental trigger, and then leaky gut. And then all that you've mentioned, those pro-inflammatory foods, just kind of, and everything else leading to the gut dysbiosis. And as Alessio Fasano wrote the book this year, which we alluded to in the last webinar, Gut Feelings, that all these chronic medical conditions probably are linked with leaky gut. And so you're getting all these things crossing over through the open gates or doors, leading to immune activation and molecular mimicry if you're genetically predisposed, and then antibodies attacking your thyroid glands. So, everything matters.
Someone had asked, how do you get the diagnosis of Hashimoto's disease? How is that done?
Well, Hashimoto's, again, is being an autoimmune disease. There's two autoantibodies you can look at: TPO, thyroid peroxidase, and thyroglobulin. Those two antibodies is what I would recommend be done. And especially the TPO will be sometimes significantly elevated with Hashimoto's. And you may not even have hypothyroidism yet, but that's the marker that you're on the trajectory towards hypothyroidism. And that those antibodies can start to appear 10 to 15 years before hypothyroidism actually develops. So look for those two antibodies.
Can you guys touch on, we had a question about your thoughts on the microbiome and intermittent fasting. And I know, Dr. Penn, I think you mentioned fasting, but can you go into that a little bit more?
Well, it's more and more recognized how we should give our gut a rest. And through there's different ways of fasting, and there's several experts out there now. There's a Jason Fung, who's a nephrologist, that's written the book, probably the first book on the guide to fasting. There's a Dr. Longo that's really looked at this, and we have can offer individuals a discount on a product that they have developed called Prolon that gives you a 5-day intermittent fasting where you're actually able to eat some, but it mimics a 5-day fast, and they don't get any profit off that, but it goes back into their research. And so why do you want to even fast? Well, we know the religions over time, including Christianity. In fact, we know that person of interest fasted for 40 days and 40 nights and then got his 3 temptations. But fasting does some fascinating things to our body. And as I mentioned, there's these guilds or generations of bacteria. And the first generation feasts when we feast, and then they produce metabolites that feed a second generation when we're fasting that has some health benefits that we might not otherwise get if we're always eating. And then the third generation is fed by the second, and so on. So there are benefits. Plus, our body is a hybrid car. We either burn dirty fuel, which is sugar, or we burn clean fuel, which are ketones. And if you're going to have a metabolic switch, and insulin is a driving hormone here, either insulin is storing fat or it's burning fat. And to be able to burn fat, you need to switch your hybrid car from sugar to ketones. And you can only do that through, well, one main way of doing that is through fasting.
I agree with everything you said, Dr. Penn. And another way of thinking about this is, remember we said we used to think bacteria were the greatest threat to human health, and now we know they can provide the greatest benefit when we take care of them. We used to think that 3 square meals a day was the greatest benefit to human health, and that may not be the case, in fact. Throughout history, not just for religious purposes, but by necessity, human beings often did not have enough food, often enough to eat 3 meals a day or even every day. So we had to be designed to be metabolically effective, even if we went without eating for quite a long time. So that's the evolutionary basis, if you will, of why we benefit from intermittent fasting. But on the physiological level, every time we eat, the mitochondria in our cells have to use those energy sources to create ATP, to create our energy sources. And in doing so, they do create a mildly oxidative and inflammatory state, which lasts probably in the range of 4 to 6 hours. So, for example, if while you're awake, you're eating every 4 to 6 hours or so, you're never really getting out of that inflammatory state. And this is why the prolonged periods of fasting are so beneficial for the mitochondria function in our bodies, which has so many widespread effects. So, what happens in the mitochondria also doesn't stay in the mitochondria. Those have systemic effects. So that's the basic link. But yes, prolonged periods of going without food are no doubt very beneficial.
Yeah, one of the first steps I have my patients take, just simply document when you start eating in the morning and when you stop eating in the evening. And they're always so surprised that they're eating like a 14-hour period of time, maybe 16 hours, waking up during the middle of the night. So I tell them, you kind of dampen this back, start cutting back over a 6-week period of time, and try to get that eating period down to maybe a 6 or 8 hour window, at least 12 hours within that eating window. And they come back saying, there are some benefits with this. I can do it, but you got to go low and slow if you haven't been doing it.
I do want to maybe talk just briefly about the sugar, because we're addicted to sugar. Yes, we are. I kind of feel like you're already going to answer it this way. Go low and slow on, don't just do like a fast cutoff because you might not stick with it. But any quick tips or suggestions about how to cut back from sugar?
I always like to start with beverages. What beverages are the patient eating, no matter what kind of dietary intervention you're hoping to make, because so many beverages are empty calories, for one thing, and beverages can be modified pretty easily. So if you can get a patient to drink water with a little lime juice or lemon juice in there, that tastes sweet. A lot of foods with cinnamon will taste sweeter if you can, if it's appropriate to add cinnamon. So there's some spices that can substitute for sugar. And, some people do have to go low and slow, but add spices, spike your water. That's a great place to start. And the other thing is, whenever you're eating a food, keep it in your mouth as long as possible and savor it. And many foods begin to have a sweeter flavor the longer you keep them in your mouth. So that's another thing that's a good idea. It also works on your satiety levels, and you end up eating less food and fewer calories if you really do slow down and savor your food and keep it in your mouth for quite a long time.
I agree with everything Dr. Luby has said. As you've implied, Rachel, we're hardwired for sugar. If you take rats and addict them to cocaine and then give them cocaine and sugar water, of course, they choose the sugar water. And so we're hardwired. And so I think the idea of just look at the beverages first and don't switch to something with artificial sweeteners, because that can harm your metabolic system. Look at, water with, again, some lemon, as you mentioned, Look at some of the soda waters that have no artificial sweeteners or added sugars. And then just kind of damp it back low and slow. And then you can start looking at your desserts next and substituting some fresh fruit, perhaps with some fermented yogurt or kefir. Like I had some excellent wild blueberries last night with my goat kefir. Excellent.
And one final comment there is have your patients do this with a buddy or as part of a group visit is even better. When somebody tries to do this alone, that's a lonely road. When you're cutting out sugar together with somebody, you can support each other. When you're doing it in a group, it gets to be a real team activity and you get much better outcomes. So that's a good clinical pearl for your office systems. Get a buddy, get a group.
That concludes the online education. I would like to thank all of our presenters for their experience, knowledge, and expertise. Be sure to subscribe so you're up to date with all of Nebraska Methodist College's continuing education offerings. Thank you.